All right, so I'm here. So what we are going to do first is to install the tools that we'll use for this exercise. So with the installation, you can use Mamba or Micro Mamba. Again, instructions for installing these tools can be found in the description box. I'll use Mamba, by the way. So this is how I'm going to do it. So I'll create an environment and install the tools. I'll say Mamba create -n and I'll say bacteria. That is the name of the environment. I'll also indicate the channel. So I'll say -c bioconda -c forge -c defaults. And then I'm going to list the tools that I will use. So with the tools, we are going to use nanoplot. This will be used for quality control of the raw reads. We will also use filtLong. This will be used to filter the reads. And then we are also going to use fly for genome assembly. I've covered an entire tutorial on that. We are also going to use fast to evaluate the genome assemblies. We are also going to use bandage. This will be used for visualizing the genome assembly. We are also going to use star AMR. This will be used for plasmids and then antimicrobial resistance genes detection. And then we're also going to use Rocker. This will be used for genome assembly. So these are the tools. Now there is another tool that has not been listed here. We are going to install later. That is JBrowse. So we are going to install that when we are about to use that. I tried initially, but I realized using Mamba to install, I was not able to open it. Or let me just say the tool was not working to my satisfaction. So I thought I will install using another approach. So for the most part, we are going to install the tools using Mamba. Let me just minimize, or let me just say, let me change the size of the text so that everything is one line. Everything is on one line. So I'll just do this so that you can all see everything. So these are the tools. So these are the commands. So just make sure to get them. So that's it. Okay. So let's install the tools now. So I'll hit the enter button to start. Okay, so Mamba has started its work. Okay, so let's type Y for yes and hit the enter. So once we confirm, Mamba will download all the necessary packages, configure them for us so that we can use them. Okay, so let's wait for Mamba to complete the installation. Okay, the tools have been installed. So we are now going to activate the environment. So we will use this command to do it. So let's clear the screen first and activate the environment. So I'll say Mamba activate bacteria. So this will activate the environment for me. So now we can start the exercise proper.